Hello, my name is Georgia Herr and I'm an environmental engineering student. My project is titled Total Oxidizable Precursor, or TOP, Assay for PFAS Analysis, and my project director is Dr. Terry in the Civil Engineering Department. So first, I'll provide some background on what PFAS are and some PFAS analysis methods, and then I'll talk about my work this semester on the TOP assay for PFAS analysis. Finally, I'll talk about where the project is now and some of our future goals. So firstly, what are PFAS? PFAS refers to perfluoroalkyl and polyfluoroalkyl substances, which is a group of chemicals that is characterized by a carbon-fluorine bond. This carbon-fluorine bond is incredibly strong and as a result does not break down naturally in the environment. This means that these chemicals are often referred to as forever chemicals and are an important issue in water treatment. As you can see in this graphic, PFAS are found in a wide variety of environments. They're often used in consumer products, food packaging, and facility activities, and as a result, they have found their way into drinking water, food, and waste sites. Now, the reason PFAS are such an important issue is they can have serious health effects in humans, including affecting our thyroids, immune systems, and reproductive health. Given how widespread PFAS are in the environment and their resistance to degradation, they can be a serious public health issue. So, how do we test for PFAS? There are a variety of testing methods available, which each paint a different picture of the total contamination. Since PFAS refers to such a wide variety of different chemicals, using multiple methods for testing can provide the best possible image of the total contamination. The most comprehensive method available is total fluorine screening, which screens for the presence of the element fluorine as a possible sign of PFAS. This can detect all different kinds of PFAS, but does not provide any specific information on the types of compounds present. For specific testing, targeted PFAS testing is available for a handful of specific compounds, but this is only a few of the thousands of PFAS compounds that exist. Now, in between these two methods, in terms of specificity, is the Total Oxidizable Precursor, or TOP assay, which has been the focus of my research this semester. This assay finds these hidden PFAS that are not detected in targeted PFAS testing by chemically converting them from precursor compounds into terminal PFAS compounds that can be detected in those tests. This image provides more detail on how this process works. So to measure PFAS in targeted testing, an analysis machine known as LC-MS is used. The LC-MS can pick up on terminal PFAS compounds such as PFCAs and PFSAs, as well as a few of these chemical precursor compounds. However, as you can see in this image, there are still a lot of unknown compounds, shown here by the unknown precursors, that the LC-MS is not able to analyze in the sample. However, when the sample undergoes the TOP assay, these chemicals are oxidized using a heat and alkaline activated persulfate chemical into terminal PFAS that can be detected by the LC-MS. You can see on the right of this image, after the assay is performed, most of those unknown precursors turn into PFCAs, which can then be detected. So the overall objective of this project is to provide insight into the total PFAS burden in a sample beyond the targeted compounds through a TOP assay. The three sub-goals of this project are to first create a method based on previous TOP literature, and then apply this method to PFAS-spiked deionized water. Finally, after perfecting this method with DI water, we hope to apply the method to an environmental sample. So the TOP assay method for PFAS was originally introduced in 2012 by researchers Erica Hautz and David Sedlak, and since then, methodology has remained pretty consistent across the literature. The key method is to use a strong oxidizing agent, typically a potassium persulfate, to convert the PFAS precursors into terminal PFAS chemically. In the method that we have implemented this semester, we have used a few small differences. Our first difference is using a smaller solution size for our samples, using only 10 milliliters instead of 125 milliliters in the original method, using a minor temperature variation in the reaction step due to some of our instrument constraints, and finally using a different PFAS standard in our samples. The PFAS standard is the PFAS that is added into the sample that is later analyzed using the LC-MS, and for our method we have been using EPA method 1633 compounds, which is a mix of 40 different PFAS compounds containing both precursors and terminal compounds, which has allowed us to analyze the reaction of multiple precursors at once in a single assay. In the original method, only precursor-specific standards were used, which only contained one specific PFAS precursor chemical. Our method steps are outlined here. First, we add PFAS solution to 5 15 milliliter tubes, 1 control tube, 2 thermalized control tubes, and 2 TOP assay tubes. Then to our assay tubes, we add potassium persulfite, which is our oxidizing agent, along with sodium hydroxide to create basic conditions necessary for the reaction. We then fill all the tubes to 10 milliliters with deionized water and heat our 2 treated and 2 thermalized control tubes at 80 degrees Celsius for 6 hours in a water bath to allow the reaction to occur. After the reaction step, we cool all tubes to room temperature and then add hydrochloric acid to our treated tubes to neutralize their pH. Finally, the samples are ready for analysis using the LC-MS instrument. You can see the method further visualized here. We start with our EPA 1633 standard PFAS solution, which, as I mentioned, is a mixture of 40 different PFAS compounds at known concentrations that can be measured using the LC-MS method 1633. This solution includes multiple precursors and terminal compounds, which allows us to analyze multiple precursors during a single assay. Our samples are then added to our water bath, which provides the warm conditions necessary for the assay oxidation to occur. Finally, we do our analysis using LC-MS, or liquid chromatography mass spectrometry. Liquid chromatography is the separation of compounds based on their interaction with a stationary and a mobile phase, and then mass spectrometry ionizes these compounds and measures their mass to charge ratio. By combining the LC and the MS, the LC-MS is able to identify and quantify the PFAS compounds that are present in the sample. For our initial work this semester, we have completed 3 full attempts of the TOP assay and unfortunately have not been successful. Our LC-MS analysis results have not shown a decrease in precursors accompanied by an increase in terminal PFAS in the TOP compared to the untreated samples. In our second and third attempts, we have been troubleshooting, first with our pH levels by ensuring our pH remains high enough throughout the entire reaction for the reaction to fully occur, and also our heating step by ensuring that our water bath remains at the correct temperature the entire 6 hours. You can see in this image on the right some observation of our TOP assay samples in that water bath. Secondly, we've collected environmental samples from local water and wastewater facilities in the Tuscaloosa area to hopefully apply this TOP acid method to once we are able to get it successfully working with deionized water. So our major future goal heading into spring 2026 is to refine the TOP acid method for success, first with our DI water samples, and then eventually apply this method to our environmental samples. After this, we hope to create a standard operating procedure for the lab group to use in future TOP assay implementation. It will be very helpful to have another PFAS testing method available to the lab group. Thank you. Please tell me your questions.
So I understand that your TOP assay method works for like sample substances outside of the body. Would it be like too wild to think that there would be a way to detect like how much PFAS we've ingested ourselves?
Yes, so this research specifically is only looking at water samples, but that has definitely happened in the field. People have used biological samples. In fact, in the lit review for this project, some of the literature we reviewed was applying this TOP method to blood samples.
Why was the method designed for a smaller sample volume?
So the major reason for that, using only 10 milliliters of sample instead of 125 milliliters, was to save on resources. PFAS standards are typically purchased 1 milliliter at a time, and that cost can add up very quickly. So using sort of as little as possible, especially when you're just setting up the method and not using it for a specific research project yet, is sort of more economical.
So are there PFAS precursors that are resistant to the TOP assay?
Yes, there definitely are. So going back to this image, you can see that there are those not oxidizable precursors after the TOP assay. And it is very important to understand that this is not going to be a one-size-fits-all for all PFAS precursors, since PFAS refers to such a wide range of chemicals. That can be a big difficulty in PFAS analysis, is that each individual PFAS is going to have unique chemical properties.
Do we have PFAS regulations in the U.S.?
Yes, we do. So for drinking water, in April 2024, the EPA released the first round of PFAS drinking water regulations, which regulated 6 PFAS. So we're currently in the period of sort of initial leading up to compliance enforcement for those. But however, in May of 2025, the EPA announced that they are intending to review and revise these regulations. So although these regulations are definitely in flux right now, we do see sort of a movement in the U.S. towards more monitoring and more regulation of these chemicals.